Triglycerides, also called triacylglycerols, are fatty acid esters of glycerol and are highly hydrophobic or water-fearing. Because of this, they must be processed before they can be digested, absorbed, and metabolized. Because processing of dietary lipids occurs in the small intestine, the water-soluble lipases, enzymes that hydrolyze triglycerides, which are found in the stomach and in the saliva, are not very effective. In fact, most dietary fat arrives in the duodenum, the first part of the small intestine, in the form of fat globules. These fat globules stimulate the secretion of bile from the gallbladder. Bile is composed of micelles of lecithin, cholesterol, protein, bile salts, inorganic ions, and bile pigments. Micelles are aggregations of amphipathic molecules, which are molecules having a polar region and a nonpolar region. The nonpolar, hydrophobic, or water-fearing ends of the bile salts tend to bunch together when placed in water. The hydrophilic, or water-loving regions of these molecules interact with the water. So you can see on the periphery we have all of our water molecules. I won't circle them all, but these are all water molecules. And then in our triglyceride, you can see the glycerol backbone here. This is the glycerol backbone. And then we have one fatty acid chain attached here. So this is a fatty acid ester bond. And then we have another one here. And these long lines here are representing the hydrophobic tails. And then, of course, we have the hydrophilic portion here. Bile salts are made in the liver and stored in the gallbladder, awaiting the stimulus to be secreted into the duodenum. The major bile salts in humans are cholate and chenodeoxycholate. Cholesterol is almost completely insoluble in water, but the conversion of cholesterol to bile salts creates detergents, whose polar heads make them soluble in the aqueous phase and whose hydrophobic tails bind to triglycerides. After a meal is eaten, bile flows through the common bile duct into the duodenum, where bile salts emulsify the fat globules into tiny droplets. This increases the surface area of the lipid molecules, allowing them to be more easily hydrolyzed by lipases. Much of the lipid in these droplets is in the form of triglycerides, which are fatty acid esters of glycerol. A protein called chylipase binds to the surface of the lipid droplets and helps pancreatic lipases stick to the surface and hydrolyze the ester bonds between the glycerol and fatty acids of the triglycerides. In this process, 2 of the 3 fatty acids are liberated. And so you can see in our triglyceride, we have our 3 ester groups, and only 2 of these are cleaved to leave a monoglyceride and 2 free fatty acids in this case. In this process, 2 of the 3 fatty acids are liberated, and the monoglycerides and free fatty acids produced mix freely with the micelles of bile. These micelles are readily absorbed through the membranes of the intestinal epithelial cells. Surprisingly, the monoglycerides and fatty acids are then reassembled into triglycerides that are combined with protein to produce the class of plasma lipoprotein called chylomicrons. These collections of lipid and protein are secreted into the small lymphatic vessels and eventually arrive in the bloodstream. In the bloodstream, the triglycerides are once again hydrolyzed to produce glycerol and free fatty acids that are then absorbed by the cells. If the body needs energy, these molecules are degraded to produce ATP. If the body does not need energy, these energy-rich molecules are stored. Fatty acids are stored in the form of triglycerides. Most of the body's triglyceride molecules are stored as fat droplets in the cytoplasm of adipocytes that make up adipose tissue. Each adipocyte contains a large fat droplet that accounts for nearly the entire volume of the cell. Other cells, such as those of cardiac muscle, contain a few small fat droplets. In these cells, the fat droplets are surrounded by mitochondria. When the cells need energy, triglycerides are hydrolyzed to release fatty acids that are transported into the matrix of the mitochondria. There, the fatty acids are completely oxidized and ATP is produced. The fatty acids provided by the hydrolysis of triglycerides are a very rich energy source for the body. The complete oxidation of fatty acids releases much more energy than the oxidation of a comparable amount of glycogen.